So we have created a Google form where you could submit your question and we'll try to address tomorrow with the appropriate answer. And each of these tools that we'll be talking about in the next 5 days, we have many other alternative software too. But keeping the freshers or the beginners in mind, I'll be using the graphic user interface softwares only. These are good enough to work, but if you have a larger data set or an advanced cross, we might need to change gears to a new software. But the basics hold good for any of these things. Our idea of a workshop, which I have been giving from the last 10 years, is end of the workshop, everyone should feel comfortable working with this again, and they should feel it's simple rather than, oh, this is so complex, let me stay away from that. We never want that. So we'll try to be simple so that you'll be able to understand any paper you read about that. At the same time, you'll be able to work with your own data. So with that, I and Divya would be organizing this. And thanks organizers, the patrons and the organizing committee of Vignan University for providing this opportunity. So as written earlier, we'll be having a 5-day. Today we'll just go, as I said, we'll be going a bit slow, trying to explain the concept of quantitative genetics. Tomorrow we'll have a demo on linkage mapping, and then a demo on QTL mapping, then a couple of days on GWAS. During GWAS, we'll try to explain LT, what population structure, kinships, what all go into GWAS. We'll be considering them and we'll try to explain with examples. So these are the softwares we'll be using for our demo purposes. For linkage map, we'll be using a software called as Carthagene, which was developed in France in early 2000s, but still holds good. Still, it's a GUI software, meaning it's just click button, no command based, making it easy for us to repeat things. And here I have provided all the links where you could download it. And this has a prerequisite of software called as Active TCL that has to be installed prior to Carthagene. And this works good for Macs or Windows. And for QTL mapping, we'll be using a software called as QGene. And here it needs a Java JDK as a prerequisite. And for association mapping, we'll be using a software called as Tassel, and Java is the prerequisite for this too. If you want to repeat any of the work we do, we'll be providing the example data sets in the folder, which I'll show you in a minute. You better install all these softwares before tomorrow so that you can simultaneously work with it or work with this after our presentation. And some other softwares that we'll be using to organize our data is one is called as TextPad. It's nothing but an extension of Notepad by a different company entirely. That's really good for organizing our outputs and inputs of these different softwares. And we need a Microsoft Excel in case, since it's a paid version, a good alternative is called as WPS Office, which is similar to Microsoft Office. It has Word, PowerPoint, and spreadsheets. It's really good. That's what I have been using over years. That's a good starting point to do some of our work or organize our data. I suggest having these two on your computers too. And this is the website. I will be uploading all our slides and data in a GitHub repository. If you go to github.com and just type my name without any dots or any special characters, you should see a Quant Genomics Workshop November 2020. It should have all of it. We'll have the data at least a couple of hours before the workshop, and we'll have all the slides immediately after the presentation. And this is a Google Forms site which we'll post in the chat window too, where you can submit your questions, your name, email address, and the questions you have. If they're relevant to a wider audience, we'll try to address it first thing in the morning, tomorrow morning. But if it is just relevant to a specific thing, we'll try, or with time restrictions, we will try to respond to each and every question through email. That's the reason we have been asking emails for. So with that, We just want to introduce ourselves first, and then we can go further. Bobby madam introduced me as a senior scientist. Thanks, Bobby ma'am. And right now I'm a senior scientist working at HudsonAlpha Institute for Biotechnology at Huntsville, Alabama. Briefly, these are the areas I have been working in the last 15 plus years, starting from genetic mapping to various aspects of genomics. Mostly to generalize, I work with a combination of genomics, bioinformatics, and biostatistics. So based on whether we apply to DNA, RNA, or epigenome, we have different names or different omics concepts. So these are different concepts I worked with. So a brief summary, I have, including my PhD, I have 15 plus years of research experience, about 5 plus years of teaching, mostly graduate level, and a few undergrad labs. I have close to 50 research articles, 47 to be accurate that have been accepted, and 3 under initial review process. I have a book chapter, and I supervised around 15 master's PhD students. I have around 30 plus collaborations active and still going. And I did so far around 10 workshops. Last couple of years back, I have been to Morocco and Tunisia just to do a workshop on quantitative genomics at ICARDA, which is more like our ICRISAT. And I have around 60 plus posters and presentations. Just a brief outline. You might feel bored. The idea is to give you an idea that the work we have been doing for years, we're presenting it in the right way. So my bachelor's was in agriculture from Bapatla. Then I did my PhD in genomics and bioinformatics at North Dakota State University, USA. So basically this university is located in Fargo, North Dakota, where the temperatures are less than 0 for about 4 months. And the coldest feel approximately like minus 40 degrees centigrade. And we have about 50 inches of snow for about 4 to 5 months in winter. So the first question that comes into mind is, how come I did a PhD there in this cold weather? And at the same time, why did I end up there? So before I go with that, this is just an outline. This is how our winter main, this is a highway, how it looks like. And this is the typical starting of a day, scraping the car glasses and cleaning up the car before going to office or work. And we have about 50 inches of snow throughout the winter. And this is our university path towards our department, where the footpath is clean and you have snow dumps on either side of the walkway. So the question most people ask me is, how did you end up in that cold traveling from India? So the main reason is North Dakota leads US because it is the highest producer of many crops like beans, canola, flaxseed, honey, durum, spring wheat. Just a few of these are listed here. So it's one of the high agriculturally high production agriculture state. So we have a lot of research and lots of funding opportunities available within the state. So that made me choose North Dakota. Just as an outline, each year we have approximately 10 billion economic impact. And here the farms, the entire state has only 26,000 farms because the average holding of a farm is around 5,000 to 10,000 acres. And here, North Dakota is third in the nation for producing barley and sugar beets, and second in the nation for producing sunflowers, beans, lentils, others. And about 90% area of North Dakota is used for agricultural production, highest compared to any other state in the US. And on the top of it, it has the highest number of bee colonies, and one major oil source in US is canola, and 86% of canola is just from one state, and 85% of nation's flaxseed is also from this state. So in North Dakota, we grow different crops: canola, soybeans, wheat, dry beans, sugar beets, barley, potatoes, you name it. Many major crops in the US are grown here, and they have at least 15 to 20% or even 80% production capacities. And my PhD work has been published, And just to show you, this was my first project I worked with, developing a linkage map for common bean, which we have been using over years for either whole genome development or many other QTL analysis. And I had like 5 papers. One was based on linkage map, one was based on syntenic comparative genomics, association mapping, population genomics, and demography. So these 5 papers make up my PhD thesis. And after that, I started as a research scientist in the same university with the same supervisor, where I mostly worked with common bean and soybean. And there I was working with variant discovery, interrogations, and QTL analysis, methodologies for higher marker creation, association mappings, and the best of all, I worked with a whole genome of common bean or Phaseolus vulgaris, which was published in Nature Genetics. And as you could see, I'm the third author. The first author, Jeremy Schmutz, is my present supervisor. Philippi McLean is my previous supervisor. And I did majority of this work. So this includes whole genome assembly, annotation, and various population genomics aspects. After that, I started as a senior scientist working on computational biology at HudsonAlpha Institute for Biotechnology. Briefly, this is our institute, a sky view. As you could see here, this is our this is a DNA shaped park which is approximately 1 mile in length, where we used to have charts and walk after lunch or something. So the idea is like we had 2 strands of DNA. One is with the rocks and the other is a plain paved path. And along the path, you have all the genes in human genomes that were discovered. This is how the interior of our building. Briefly, we're a non-profit research institute doing research, education. We train school kids all the way till doctors, PhDs, scientists, others, based on the necessity. And we do knowledge sharing with the bio entrepreneurs. Our building itself is a combination of a non-profit organization with around 40 different companies. So the overhead of this entire building is mostly run by the rent or other resources provided by the 40 different companies. So it's first of its kind, where a combination of non-government and this private or bio entrepreneurship in the same building, which has been replicated in Europe later on. And we have bioinformatics and sequencing services, and we are the pioneers in genomics medicine. So these are the main research areas at HudsonAlpha. We do some basic research in humans, pediatrics, neurological disorders like Alzheimer's, Parkinson's, many others, different forms of cancer, genomic medicine, as I'll be mentioning in a bit. Agri-science is something that we are working with, where we apply genomic knowledge to agriculture and bioenergy, immunogenomics, and developing tools and other things for computational biology and bioinformatics. We're about 10-12 groups of researchers with around 200 staff members. Genomic medicine, we call it as the Smith Family Clinic for Genomic Medicine. We are the first clinic to offer genomic medicine. So the basic idea is, if a doctor is not able to diagnose a disease based on the symptoms, they come to us, we sequence the whole genome of that human being along with their parents if still necessary, and we try to relate the known variants or known diseases from the genetic database so that we can provide the doctor and the patient necessary remedies. And we want to give the best positive medical experience possible. So far, we have screened about 400 patients in about 4 years, where 2/3 were adults and 1/3 were children. And most of the symptoms we identified were developmental delays and intellectual disabilities, neurological issues, immunodeficiencies, which by ways of symptoms cannot easily be identified, and many other unexplained physical symptoms, unexplained heart abnormalities. And when we talk about genomics, the first thing that comes into mind is the initial paper on human genome, and that was published in 2003. And our institute's link with the human genome is that Dr. Myers led a team at Stanford Human Genome Center where they contributed more than 10% of the data. And with him were working Jane Negrimwood and Jeremy Schmertz, who assembled 3 chromosomes, which led to the start of genomics in humans. Uh, Worth mentioning is that these are the 3 persons I was talking about. Richard Myers, who then was working at Stanford, is right now the president and science director of our institute, or he initiated all this effort. And Jeremy and Jane are my present supervisors, husband and wife. Jeremy takes care of the bioinformatics computational part. Jane takes care of the experimental and the wet lab part. And in addition, Jeremy is a plant program leader at the Department of Energy Joint Genome Institute, which is located in California. This is a little older slide, around 4 or 5 years old. Till 2016, we have about $165 million in grants and about 550 publications till 2017. So since I work with the plant genomics group, when I started working here, it was just our group, around 6 people working on the computational side. Now we have 3 other faculty members, so it's expanding. So basically, we work for optimizing crops for climate changes, increasing water use efficiencies, working with plant pathogens, fertilizer use efficiencies, and adapting crops to marginal lands or agronomics, in addition to many other abiotic stresses and other things. In terms of analysis, we work with the comparative genomics, building reference genomes, population diversity and genomics, quantitative genomics, and transcriptomics and functional assays. I work mostly with the quantitative and the population genomics. So far, we have more than 50% of plants are sequenced, whether it's soybean, wheat, rice, Arabidopsis, you name it. And our group has sequenced more than 50% of them. And most of our work has been published in all high-impact journals, Science, Nature Biotech, just to name a few. We have around 50 Science, Nature, and Cell papers just from our plant group. And out of the 12 faculty members, most of our faculty members are Thomson Reuters highly cited researchers from the last 5 to 10 years. And my work here is mostly variant discovery, where I'll be working where I work with identifying SNPs, indels, CNVs, and many other structural variants. As an example, each data set could be terabytes with thousands of samples. One of my recent switchgrass samples has approximately 1.5 to 2 terabytes of data that we use to analyze. And in addition, I work with population genomics, where I look for climatic adaptation, demographies, adaptations. I work with epigenomics, pangenomics, functional genomics, just to name a few. And these are a few biofuel crops I worked with: switchgrass, setaria, poplar, panicum. And these are other crops I'm working on right now: pecan nuts, which is a tree; zostera, which is a marine plant, stays under the ocean; and a wheatgrass; sorghum, which is mostly for Africa. This is funded by Gates Foundation, so mostly for helping African nations with sorghum. And beans, and this is sphagnum, which is a small algae type that's grown on rocks. That's good for carbon fixation and understanding carbon sequestration. And my work here so far has been published in Nature Communications, couple of them, and one Genome Biology, and recently in Nature Biotechnology as a lead author. And I have one other paper coming up in Nature where I'm a first author too. And this is my present group at the Genome Sequencing Center. And over years, about I would say around 10, 12 years, I have been collaborating and supervising many other graduate students and collaborating with faculty members. I'm fortunate to work with linkage maps, QTLs, association mapping, marker development, comparative genomics, and many others. I had the privilege to work with various aspects of genomics in different crops. So I work with diploids, polyploids, outcrossing, self-crossing, where a few methods or a few principles change for each one of them. I'm fortunate to work with all of them, so I'm more capable of working with any other thing. And some of the species I work with include around 7-8 crops: wheat, durum, canola, rice, pea, barley, oats. In addition, I work with humans, mouse, and recently I have been working with a few microbiomes, gut microbiome and soil microbiomes. These are a few papers I have published over years with my collaborators: association mappings, QTL mappings, linkage mapping, which I'll be talking about in the next few days, around 15 to 20 of them, I believe, and this expression RNA-seq analysis. In addition, I have around 4 papers in human cancer studies where we used RNA-seq and other approaches. And this is a small graph showing all the work I have been doing these years. And as always, we are open for collaborations for improving science. With that, I'll stop for today. I'll let Divya take over. And after that, we will get back to the original workshop or more the demo part from tomorrow. Today, Divya will be talking about herself first, and then she'll be talking about the concepts for linkage and recombination. Divya, you want to take over?
Yes, I'm able to unmute now. Can you hear me?
Yes, yes, Divya ma'am. Sorry, ma'am. Some
Oh,
technical.
that's okay. That's okay. Uh, Thank you, sir. So now I will take over the session. Today it's going to be like, as Dr. Sujan sir said, it's going to be a very introductory session. So it's not going to be a very detailed, it's not going to be entirely a lecture mode, because I assume that people know some concepts in genetics. So before we move on to the demo sessions, which is scheduled tomorrow, we would like to brush up some basics. And above all, I would like to introduce myself in the coming slides, actually. So let me share the screen. And so apologize if I'm going to like stop my video, actually. So okay. Thank you everyone for attending this session. So being the first day, I would like to talk about, introduce myself, my introduction and my research journey. So that's going to be the part one of the session. One second. Uh, okay. So before I move on to explain about my work, I would like to say that uh, I did my undergrad in like BTech in horticulture from Tamil Nadu Agriculture University, Coimbatore. So it's a very known university, and it's got a lot of history. 100-year-old university. So, and lots of people like MS Swaminathan emerged from such universities, uh, the father of Green Revolution we call today. And I'm very privileged to have the education, the undergrad education from Tamil Nadu Agriculture University. And thanks to all the teachers for carrying out for successfully carrying out my journey till now. And following that, like, I started doing my master's in biotechnology from TNAU again, that is Tamil Nadu Agriculture University again. And then we got this dual degree program offer, funded by Ratan Tata Foundation. So I was one of the persons selected for the funding, and I was funded for the master's program for a year at Cornell University in the US. So where I studied masters in plant breeding. So I got a dual degree masters in plant breeding as well as biotechnology. And thanks to Ratan Tata Foundation for the funding. And later, I moved on to doing PhD. After I completed my thesis in both the universities, I joined the PhD program at the University of Cambridge. So where I was selected for the Marie Curie funding fellowship. And this was actually a dream when I was doing my master's, because hardly a few people are selected around the world for a specific project, for a specific field every year. And for so long, it was a dream until I completed my master's. And I was fortunate to find a good lab and then land at the University of Cambridge. So this is something I wanted to tell people, and I found a lot of students in the list. Some people might be JRF would be in their masters, or might be in their might be in the verge of joining a PhD program. So I would like to say that you guys, if you are looking for an opportunity abroad, in the UK or in the US, kind of look for a lot of fundings and fellowships. Lots of fellowships are being given by the Indian government. If you look into the ICAR website, the Indian Council of Agriculture Research, they have lots of like fellowships and tie-ups with big universities or the great universities in the US and UK. Also, I would like to say, like, it's fairly challenging as well as not extremely impossible to apply for a Marie Curie fellowship, which is a major fellowship for PhD program, which is one of the prestigious fellowships for the PhD program that is given by the European Commission, basically. And you can also look for like other fellowships like the Commonwealth fellowships that is normally offered like every year. I saw some postdocs as well, like active postdocs and some PhD scholars in the list. And if you are interested, you guys can look for Commonwealth fellowships, and there is a fellowship offered by the Marie Curie Foundation itself for the early career researchers, or I would say like experienced researchers. So postdocs are called experienced researchers. So hence, they give you a very good package in terms of your fellowship amount, as well as it gives you a good opportunity to explore a different environment, their research projects, and entirely I would say like it gives you a holistic experience in total. So if you're applying for that, please let me know. You can always contact me for if you want to have some sort of career guidance or some sort of want to know about some fellowships in the future. So this is something like I applied like just in a casual manner for the Marie Curie fellowship, and I got so, and went through a round of interviews, a telephonic interview, and later a formal interview with a presentation, and then I got admitted into the University of Cambridge. So I had a 4-year long journey, and I'm going to talk a little bit about my project. So, in contrast to what Dr. Sujan sir did in his PhDs, and it was more into crops and more of application-oriented research. And although it's a lot of data analysis works, lots of bioinformatics work, power packed into those projects, still, like our projects in Cambridge mostly were like more basic. So we dealt with basic research and the model plants like Arabidopsis. So my department is the Department of Plant Sciences, which is in the central Cambridge. So what I did basically was I worked in a process called meiosis, and specifically, and I would like to say what is meiosis. So meiosis is basically involves between sex cells or the gametic cells. It involves a round of DNA replication followed by 2 rounds of nuclear division. So there are two different parts in meiosis: the meiosis 1 and meiosis 2. So during meiosis 1, there's a specific process takes place called recombination. So recombination takes place in a very specific stage where I don't know like how many of you have learned or been aware of the cell division process and the different stages of meiosis. Especially in meiosis, there are different stages like prophase, metaphase, anaphase, and telophase, and that has been repeated in the meiosis 2 as well. But the longest stage in meiosis 1 is the prophase 1. So, and it involves 5 different stages, starting from leptotene, zygotene, pacotene, diplotene, and diakinesis. So where recombination takes place is specifically where the arrow is marked, like in the pacotene stage, where there is an exchange of genetic material takes place between the parents. So between the homologous chromosomes especially. And the subsequent stages where the homologous chromosomes like once replicated, they are segregated, and then finally they form, finally the haploid gametes are formed at the end of meiosis 2, and again they participate, they are fertilized. Once they are fertilized, the gametes unite, and once they are fertilized, they will be forming diploid cells. And again, like the gametes can participate in meiosis to continue the meiotic process throughout. So during meiotic prophase 1, the important process I worked was recombination. So to tell something about the pathway, so I specifically worked in Arabidopsis, where this is the meiotic recombination pathway and what happens between the homologous chromosomes. So I would say like these two colors, like, one is in blue and the red. This is a maternal, this is a chromosome pair from the maternal parent, and this is from the paternal parent. And then they undergo something called double strand breaks. There is a small cleavage that happens in the presence of a variety of enzymes, and then like and then they lead to formation of recombinants and non-recombinants, especially like we call the recombination products as crossovers and the non-recombination products as non-crossovers. And these non-crossovers can come from gene conversions as well, or spontaneous mutations. And what happens during the stages, there are several stages in this process, and they are being driven by a variety of proteins. I don't want to talk about all these things in detail, but I worked in identifying proteins or genes that involve in formation of crossovers. So in Arabidopsis, the problem is if you have 250 breaks, 250 double strand breaks per meiotic cell, when it happens, only 10 forms crossovers. And of which, so that is like only 10 forms crossovers, and the rest of them would lead to non-crossovers or gene conversions. So in order to like identify what genes contribute to these recombination so that they can be manipulated in breeding programs in order to increase the recombination rate in genome wide in organisms, especially in crops, which would be beneficial for agriculture purposes. So my PhD work was, I simply developed a genetic screen, which is called a forward genetic screen, to identify genes that control meiotic recombination in Arabidopsis. So Arabidopsis is the model genome. It is only 135 megabase pairs in size. And this is the flow. I would like to say this is the method. These are the methods I used, and this is still unpublished. Because I recently completed my PhD, I graduated last year, and since then we've been reviewing into the paper, into the manuscript, and it will be shortly be published early next year. So I will be the first author for this paper. So what we did is simply conducted a genetic screen to identify genes. So earlier, for any meiotic screens, people used to identify or choose plants based on the seeds, if the seeds are sterile, or if the seeds have a different phenotype, like if they are fully fertile, or if they are contrast to being fully fertile, they would identify such phenotypes and go for cytological screens. They further identify at chromosome level by staining against different antibodies, and then they would further go for maybe a map-based cloning to identify the gene responsible for the phenotype, and also locate the distance between the genetic markers. So what we did actually was why we conducted the screen was we had more potential to isolate uh, more modifiers of recombination actually. So we basically identified mutant phenotypes that are higher or lower in recombination rate. So we used a simple method called using genetic intervals that are fluorescently tagged. So we used GFP and RFP markers that constitute a genetic interval along the chromosomes. And I used this particular marker in chromosome 3 for called 420, which is like a 5 megabase pair which constitutes a 5 megabase pair physical distance and a genetic distance of like 20 centimorgan. So when we measured recombination in the mutant populations, we kind of found like higher, so mutants that are higher or lower in recombination rates. And then we pulled out those mutants and developed a backcross mapping population and used next generation sequencing platform to identify the mutation that is responsible for the deviation in the phenotype. So this is like a pipeline where I use pipeline of my whole research, actually, like how we arrived at getting a mutant population by treating EMS, and then further I developed into like 7,000 we got a lot of plants, and from there we divided into small subsets of those large population of mutant plants. And then we actually used the help of used microscopy to select seeds that already have fluorescent markers, so that constitute this genetic interval. And then we measured recombination rate. I mean, the formula will be told later. So we just measured the recombination rate or the crossover rate in the mutant individuals, and found that a variety of individuals showed like more than 20 centimorgan and then less than 20 centimorgan. So later we had to backcross with the wild type, the parent, and then we developed a huge backcross mapping population, like nearly 600 individuals. And then we found a clear segregation of the mutant phenotype, which is only 1/4, because this is a recessive phenotype. So we found like 1/4 of the 600 individuals showing mutant phenotype, and we pulled all of those DNA together and developed a DNA library. So here you can see the pipeline in the third picture, where I crossed the homozygous mutant individual to the parent, which is the wild type, and then found the HEF1 heterozygous for the parent, as well as heterozygous for the markers, actually, which we use for phenotyping this whole population. And then in the next generation, which is the segregating population, or the F2 population, we found a quarter of individuals showing homozygosity for both the type of marker for the mutant phenotype. And then later we went for the mapping by sequencing technique, or we call as deep sequencing technique, and then we were calling SNPs that had a shift between uh, like G to A or C to T, because EMS treatment would normally would normally create point mutations. So it will have a shift from G to A or C to T. So we found a C to T mutation in a chromosome 4, and then the candidate gene was revealed later. So the next process, like the next half of my PhD project, was more into characterizing whether this gene is responsible for the phenotype or not. So we had to do a lot of background experiments, a combination of cytogenetics, etc., to arrive at this kind of conclusion, to arrive at a point where this is responsible for the recombination phenotype, and it has a specific function in recombination. Say it is involved in the beginning of recombination, or it is involved in the middle of the recombination pathway. So as I showed you, there's a flow in the pathway where the lots of proteins are involved in the pathway. So I found that these modifiers can impact a specific step in the recombination pathway to drive more recombination in Arabidopsis. So this could be a potential application in the future, if necessarily or importantly, considered and incorporated in breeding programs. So this is all about my work, and I would like to acknowledge my PhD supervisor, Ian Henderson, and Kyuha Choi, who is a former postdoc and is now a collaborator and a professor working in Pohang University, and he has a big lab, and he is looking for active postdocs. If you want to learn genomics, if you want to learn next-gen sequencing and genetics and combination of cytogenetics, you can definitely approach him for a PhD or a postdoc opportunity. And then there are lots of people who worked at different stages of my project. So my project required a lot of cytogenetics, lots of genomics, and then the analysis were done with the help of bioinformaticians. and hence the project was successfully completed, and it is now in the publishing process. So thank you for that. And then now I move on to the part 2, which is just a few concepts in genetics to refresh. So I hope if you have any questions during the session, please type it in the chat box, or please send it through the Google Forms if it is, so we would be able to get back to you. So we all know like the Mendel's laws of inheritance. I think most of us would have studied it, like we would have studied right from the school. And he basically proposed like 3 laws of inheritance. So he safely worked on pea plants, or the Pisum sativum, which is a good model, for raising a large population. And basically this guy wanted to understand how traits or characters are transferred from one generation to another. So that's why he arrived at these principles of heredity, or the laws of inheritance. So the first law which he talked about was the law of dominance, which where most of the time he meticulously counting peas, and he termed, before he actually concluded that these are responsible, like the traits are the genes are responsible for this kind of traits. He named them as factors, which you might have heard. And then he actually named as dominant and recessive characters, in fact, for the individuals. So what he said was, what he stated in the law of dominance was, when 2 homozygous individuals, like you can say like a tall short, I would say TT and TT, because each individual with each gene has 2 copies, and the alternative forms of a gene is called an allele, which you might know. And or like what do you say, when 2 homozygous individuals with 1 or TT or more sets of contrasting traits are crossed, like RRYY, the visible characters in the F1 hybrids are dominant. So initially he thought that if we mix a tall and a short plant, he would get all medium. So he thought a blending model would work actually, but it failed. So the actual results was, he found all of them were tall in the first generation. So hence the F1 hybrids were dominant, and those which do not appear are called recessive. So, and when it was self-fertilized in the next generation, I mean in the segregating population or the F2 population, he found the phenotypic ratio as 3:1. So which is like the dominant individuals where the proportion were 3, and then the recessive was only a 1/4. So when you look into the genotypic ratio, which you might have heard, you would always have like the 50% of individuals with both the copies, like 1 copy of each tall and short. So hence the genotypic ratios will be like 1:2:1. So this is a simple monohybrid cross, which I would want to talk about while explaining, because of explaining this concept of dominance here. And the second law which he talked about was law of segregation, or the purity of gametes. So what he said was, only one of the 2 copies of the factors or genes, so the genes, it was later coined as genes, enters each gamete. So the gametes participate in meiosis, and then after fertilization, the gametes unite to produce F1 after fertilization, and the F1 heterozygotes with and the F1s are heterozygotes with 1 copy of each. So which could be which could be a Y capital and the Y short. So they are entirely, it could be like a tall short, kind of, or yellow green. So these kind of traits. So the heterozygotes will harbor like 1 gene of Y and Y. For example, here the parent P has yellow color, parent 1, and the P2 has a green color, and then the gametes are formed, and the F1s will have a combination, will have 1 copy of both, and then the F2s will have a 3:1 segregation. So only 50% of the gametes will have a combination of, will have Y and Y. So all possible combinations will occur in the F2, that is a segregating, because it's a segregating population, when gametes unite at random. So, and the thing is, where Mendel actually found only like he only investigated on 7 different characters in P's, and all these characters were only single gene driven, and they were not, and he did not talk about, or he did not investigate on quantitative characters or the polygenic or driven characters. So, and he also he was the first person to actually use some statistical methods to analyze and predict these inheritance factors. And the third law, which is the most important for us to for this kind of session, for this whole week, is the law of independent assortment. So for which I have kept dihybrid cross as an example. So when you have like 2 or more different genes, for each pair of traits, say yellow and round and green and wrinkled, so, and they assort out independently into gametes of the other pairs. So the allele of one gene actually received by the gamete do not influence the other, so which is other gene actually. So the dihybrid cross ratio, like when it segregates into the F2 population, so you have like you have round and yellow and round, and then you have the green and wrinkled, and then you have the heads which both the characters. So he also confirmed whether these crosses worked, or whether these phenotypes are actually the real one, by using a test cross in the F2 population, by crossing this F1 into the with the parent 2 actually, and to confirm the ratios as correct. Like when you do a test cross, actually you normally get like the 1:1:1:1 with the parent, which confirms that it confirms the recessive inheritance of a subset of this population. So there is another question: why alleles may not assort independently at different loci? So there are some reasons that I want to say, and it could be either by chance. And the most important thing that we should note is, if the genes are different loci or on the same chromosomes and are very tightly linked together. So the second reason is the linkage. If the genes are different loci are very tightly close together on the same chromosome, then they may not assort independently at different loci. So when the genes are linked, they fail to assort independently. So the law of independent assortment was basically, it takes place during meiosis. So before Mendel actually coined about meiosis, coined about the law of independent assortment, meiosis was, they were not actually inferring about or actually talking or researching about meiosis. So meiosis, the mechanism of meiosis was later understood after Mendel postulated the law of independent assortment. So what happens during meiosis? As I said, that exchange of DNA or the genetic information takes place during stage packaging in prophase 1 of the meiosis, the first meiotic division, or the meiosis 1. So haploid gametes, as I said, are formed from diploid cells. And what happens is there's an independent assortment of genetic loci takes place. And this takes place on different chromosomes, or it can also take place on the same chromosome, provided they are far apart from each other. If the genes are far apart from each other, there is an independent assortment that would take place. And this takes place via the crossover or the recombination. So just to brush up the terms such as linkage and crossover. So what is linkage actually? So genes on the same chromosome that are linked and they fail to assort independently. So such genes, they actually segregate together in the population. And if they are beneficial genes, they are good for agriculture, they are good for breeding purposes. If there are non-desirable genes, then they are not. So most of the agriculturally important traits, they are kind of linked together and they segregate together in the population. And Linkage is based on the frequency of crossing over between the two genes. So the more the recombination frequency, actually, they are the linkage is not taking place. They are farther apart. The genes, it means that the genes are farther apart. They are not linked. So crossing over means is the reciprocal exchange of chromosome segments between the homologs or the non-sister chromatids. So this exchange of DNA actually takes place, as I said, during packaging stage. So this is a non-sister chromatid, what I'm showing here, and this is a homologous chromosome pair. And then during this chromosome crossover, that is small exchange takes place, and the bits are actually exchanged here. Here you can see the red bit here and the blue bit there. And these are the top, the left corners are like the other pairs are called the non-recombinant chromatids, and these are the recombinant chromatids. So what happens when the exchange takes place? There's a lot of genetic variation that is being produced, and that's why, even human beings, why we are all different, because there is this kind of recombination that is driving us to be different, because of the meiosis, because of the first stage of meiosis that takes place in every egg or the sperm cell. So the genetic variation is created and the gametes united random after the meiosis, and they fertilize, and then the diploid cell is formed with new combinations of alleles. And this could be beneficial, and some parts of this could be very beneficial for breeders, actually, to have a big chunk of germplasm for them to choose a plant and then use for introgressing various traits into the elite cultivars, actually. So there's something I want to talk about, linkage map. So you might have heard about genetic map or chromosome maps. They are quite the same. The linkage map is called the genetic map or a chromosome map. So what is this map actually? So a map which is based on recombination frequencies between genetic markers. So between two markers, for example, during a crossover or a recombination that happens during meiosis. So how do we measure the genetic distance, or the recombination frequency? And what is a recombination frequency? It's a measure of crossover or recombination that's happening between two genes. So that constitutes an interval, basically, a genetic interval along the chromosomes. So this indicates how much recombination is actually observed in a particular experiment. So if you have more frequency, the greater the frequency of recombination between the two genetic markers, it means that they are farther apart, actually. They are not very close to each other. And what is a genetic distance? So it's another term of recombination frequency as such. So there's a formula where people calculate between two genes, which I will show you in the subsequent slides. So what it means, it equals like 1/2 the average number of crossovers. So if the average number of crossovers is like 10, and half of that is in that region, it's 5 actually in that particular cell. So 2 genes equals 1/2 the average number of crossovers in that region per meiotic cells. In Arabidopsis, it has 5 chromosomes, and approximately in every chromosome, 2 crossovers takes place. And about 85% of the crossovers takes place, takes place according to the class 1 crossover pathway, which is called, we also call it as interfering pathway, where interference is useful for identifying double crossovers, where the crossovers happen adjacent to the two markers, actually. So basically, if you have 3 genes, double crossovers, there is a possibility of double crossover taking place. So, and what are the uses of linkage map, basically? And earlier, if people wanted to identify the genetic distance between the two markers, and if they wanted to identify the genetic basis of a particular phenotype that is different from the wild type, they would want to go for a map-based cloning, which is a very tedious process, actually. So which is a very, very laborious process and it takes a long time to identify these markers, because it has to involve a large population. And this was the basis initially to develop the first genetic map. And and nowadays, you have the help of next generation sequencing to work on, to efficiently identify lots of SNPs or markers to predict a particular phenotype, that is actually elevated by the contribution of a gene, basically. And then secondly, you can also identify lots of genes conferring a particular trait of interest, a particular agronomic trait of interest. And then it also helps in estimating like gene effects of essential agronomic traits in crops. Especially, it can estimate what to say, like the F1 effects, for example, heterosis effects of essential agronomic traits in crops. And then it can facilitate by getting more markers, by identifying more markers for a specific gene of interest. It can allow in facilitating marker assisted selections, so we can actually distinguish between genotypes in a population for selecting the best line that can be moved forward for developing a variety in a crop. So, and also linkage maps have always been useful in studying the genome structure, because like lots of information is kept on, is being fed with the advent of markers that has been identified either through the old method, like the marker, like the map-based cloning, or by the new methods like the next generation sequencing techniques. So it helps us to reconstruct linkage maps, helps in identifying the genome structure, its overall organization, and also the evolution. So, and a brief about like what are all the major steps involved in linkage mapping. So what we require basically is we need a mapping population and we need molecular markers for genotyping the mapping population. And the rest actually will be, and then later we'll be calculating the recombination frequency. Then we construct a genetic map. And the further, like the applications are like further into QTL mapping and marker assisted selection. So we will be doing a lot of this demo, and Dr. Sujan sir will be helping you tomorrow in doing in working on this linkage mapping tomorrow. And it's just like I would want to brush up what are all the requirements for the linkage mapping procedure. So we need a mapping population, and we extract the DNA. This is the pipeline basically. We extract the DNA and we do a PCR and we identify, now you can see the segregating populations here. And then, as Dr. Sujan sir said, you have softwares to actually score and code. You manually score it, and then you feed all the data into the mapping program, and then the linkage map is constructed, and you can see the position of these genes along the chromosomes and the genetic distance. So you will do all these things tomorrow, actually. In a more detailed manner, it will be explained. And developing this kind of mapping populations, you can either use a backcross mapping population where the F1s, the hybrids, are subsequently crossed to a recurrent parent, and then a backcross population is developed upon selfing the first round of backcross population. And then you can also use the segregating F2 population or the RILs that are actually descended from every single plant, every single F2 segregating plant. So, and also like people also use double haploid populations. And so these are the kinds of populations that can be developed. And then further, we look into the different types of markers, actually. So people use either mostly they use codominant markers these days, especially a lot of SNPs and SSRs have been used a lot. Predominantly in India, people have been working a lot on SSRs, especially in rice. And so there are different types of markers, especially codominant markers and the dominant markers. So dominant markers, where when you design primers in a specific region or a specific segment of a plant, it will distinguish genotypes. It can distinguish homozygous from heterozygous genotypes, actually. So whereas in a dominant marker, it will only give you one dominant genotype. It will only distinguish a dominant genotype, whereas it will not distinguish the recessive genotype from the dominant. It will not it will not identify the presence of a recessive genotype and the heterozygous genotype, which has each copy of the dominant and the recessive allele, actually. So one example is the RAPD, which is the random amplified polymorphic DNA. So these markers are like, it's like normally used in the beginning of like before people wanted to explore into different markers in the earlier days. They would want to use like RAPDs in the beginning for crops and to narrow down the phenotypes. So what RAPD is actually like, basically their random segments are basically amplified using a single primer of an arbitrary nucleotide sequence, actually. So when it comes to codominant markers, as I said, here you can see like, you can distinguish between parent 1 and parent 2. And this is the F1 that has both the strands, both the bands of DNA. And then this is a segregating population where you can distinguish between heterozygotes from recessive and the dominant parents. So parent-like phenotypes. So that's the advantage of codominant markers, to distinguish between genotypes. And normally in marker-assisted selection, which has been used for breeding programs, we use a lot of these codominant markers, actually, like SSRs or SNPs and caps or decaps. If we know the mutations already, caps and decaps are normally used for identifying the known mutations that are very specific to a gene of interest. If we are looking for a specific gene that is conferring a specific trait, say heading date in plants, in rice, and you have these markers responsible for the heading date, and it will help us to screen through the process, and we can develop some caps and decaps markers which has only a SNP difference between the wild type and the population and the mutant. So, and basically we can identify caps. We can identify the individuals in the population that differ only by a SNP, actually. Either they may look like wild type, which is or they may look like the mutant phenotype, which has an altered heading date, for example. So heading date is basically the time of flowering. So which is very much related to a photoperiod in plants. So in rice, people normally look for a short heading date because they wanted to complete the cycle of harvest, and they wanted to increase the productivity of rice throughout the year. So hence people were working on heading date in my lab. So they were using a lot of these SSR markers, and they also created some screens where they identified the genes responsible for the heading date, and they kind of developed some caps and decaps markers as well to identify the difference between the variant phenotypes. So caps and decaps mean it's cleaved amplified polymorphic sequence, or the derived cleaved amplified polymorphic sequence. Basically, by using these caps, it creates a restriction site, actually, because when we design markers, we add the restriction site into it, and then once it is mixed with the DNA, and then a PCR is done, and we use the specific enzyme to cleave at a specific region where the mutation is created. So where there is a mutation, there will be a cut in the fragment. So where there is no mutation, you will have like a non-cleaved fragment. So you can basically distinguish between the cleaved and the non-cleaved fragment, and that's how you distinguish between genotypes of interest. So SSRs are microsatellite polymorphic markers, as I already told you, being predominantly used in rice. There's a huge, lots of markers for specific traits in rice, basically, and you can find in the Grameen database with all these SSR markers being updated. So these are called the simple sequence repeats, and you can also find like even 10 alleles per locus. They are highly polymorphic, and they can basically distinguish homozygous individuals from the heterozygous individuals falling from the same origin in a population. So they can and whatever that is found outside the SSR repeats, usually they are the conserved region, and people basically use these conserved regions for developing markers for genotyping. So the lesser the repetition of these SSRs that you find, per locus, you may expect, you may actually see that they may be very highly polymorphic. Their polymorphism level will be like very high if they are lesser in repetition per locus. So an AFLP and RFLP markers are basically like, RFLP was originally used, basically you distinguish genotypes based on the restriction fragment lengths. So when the fragment lengths are digested using restriction endonucleases, then you may find difference in lengths, and that distinguishes between the A genotype from the B genotype. And AFLP is an amplified fragment length polymorphism, which involves an adapter ligated PCR followed by an RFLP. So it's a combination of PCR and RFLP where you can actually distinguish, again, you can distinguish between homozygous and heterozygous individuals. So hence, like RFLP, AFLP, SSR, SNPs, basically, they are like single nucleotide transitions or transversions. So transitions are either between C to T or G to A, and transversions can be C to G, something like that. And and then you can identify this, and basically these caps and decaps markers are themselves being used to identify or detect SNPs in a population. The larger version or the new version of identifying millions of SNPs are the next generation sequencing, which I will be talking about in a minute. But you can, what do you expect, like according to different populations, what do you expect the segregation ratios would be? So if it's an F2 population, for a codominant marker, say an RFLP or anything, or SSR, you will find a ratio of 1:2:1. Whereas in a dominant marker, because it does not even detect like recessive individuals or the het individuals, it will be 3:1 ratios. In a backcross population, it's actually 1:1, because it's only going to be between because it's only a population that's developed from a hybrid, like a HEF1 and a recessive parent. So the ratio is going to be like 1:1. Whereas in dominant markers, again, it's going to be 1:1, because again, hets are going to be showing or reflecting only dominance. So it's going to be 1:1. So in a recombinant inbred or a double haploid, again, it's 1:1. And for a dominant marker, it's 1:1 again. Because recombinant inbreds are, again, they are inbreds developed from F2 populations, and upon self-fertilization. So the ratios, the ratios between dominant and the recessive is 1:1. So some advantages of these molecular markers, just to say, is like so the more markers we identify in a population, it can help us to refine the construction of genetic maps. We can put in more information and fill in the gaps in the genetic maps. Because earlier, when we did map-based cloning or the older methods, like there were larger gaps. People were not able to retrieve all the information or get all the informations. So this will, like the more introduction of markers will help us to reconstruct the genetic maps, and then more genes of interest will be identified. And the work of geneticists like me is like, we actually characterize those genes responsible for specific phenotypes or the specific trait. So, and later, and the next thing is people would want to find, by using these molecular markers, we can find the effect of this hybrid vigor. So this is a term that you might have heard, like actually hybrid vigors are actually the F1s, which are much better than their parents. So we also call them as heteroses. So to find the effect of this hybrid vigor in the population, so molecular markers are extensively used. So, and then as I said, like how we use molecular markers in genotyping different populations like the backcross populations or the F2 segregating populations, it can distinguish individuals between heterozygotes and homozygous, like heterozygotes with one copy of each of the genes and each of the gene copies, and then the and the homozygous have two identical copies of those genes. And we can also like do some comparative genomics between different species, like which share, for example, sharing a common ancestor or something like that. We can actually detect the variation between species using the molecular markers. So it's a great advantage. And we also utilize these molecular markers widely in backcrossing to identify the gene of interest. And we also genotype these backcross populations while we are introgressing these specific genes into the populations. So a specific disease gene of interest into the population, we actually we actually genotype the whole population to for the presence or the absence of the introgressed gene. So and again, as I said, linkage mapping is one of, like, using of molecular markers can help in constructing the linkage map. And then different populations like QTL mapping, GWAS, different association mapping populations, which help us to, like, QTL mapping is something that we will be discussing day after tomorrow. And I think you might be aware that we actually locate the genes of interest that confer quantitative traits. Most of these quantitative traits are like, say, hair color or something like that in humans, like they are not contributed by a single gene. For example, they might be contributed by different genes, or even the abiotic stress genes. They are quantitative in nature in plants. So it helps, QTL mapping helps us to locate the major genes that confer these quantitative traits in the population. And the next technique is GWAS, where Sujan sir will be talking more about that. So, which is going to be, which is a very powerful technique actually to study natural variations. So through GWAS, actually, there's like millions of SNPs are being produced, and this itself is a repository of identifying or developing new markers for the traits of interest. And then the marker assisted selection, as I said, which is very useful for genotyping populations. So, like earlier we were using molecular markers. We were using map-based cloning techniques to identify the distance between the markers and the markers conferring the trait of interest. By using a large population and then developing back clones and then assembling those back clones to construct the genetic map. But this is a very strenuous process, very time consuming, and it's not very efficient in the current scenario where we have so many genomes to work with. And so next generation sequencing it's a groundbreaking technique in the recent times, and where millions of SNPs are being produced at very low cost. So again, I said, like, they have a variety of applications in GWAS and in identifying different mutations for a specific gene of interest, and also in linkage mapping. So, and I think you will be aware of the different platforms developed by the companies like Illumina, Roche, or PacBio. So they have their sequencers, and they have a pipelines where the vet biologist basically construct a DNA library or any genetic material, which could be an RNA library, and then they sequence it using these Illumina or Illumina platforms, etc. And then the work of bioinformatics come into place. So people analyze it, assemble the genome, and then annotate the genome, and then provide all the genetic information required. And then you can derive a lot of SNPs that are being called during this process of sequencing analysis, and that can be utilized for various purposes, like, as I told you, linkage mapping or MAS or GWAS, etc. And the other technique that is coupled with next-gen sequencing is the genotyping by sequencing, which we largely do in our labs, actually, for mapping crossovers or recombination in the segregating populations or F2 populations. So this is also a technique which is like a modernized version of the NGS, which is also used to identify millions of SNPs, actually, a greater magnitude of SNPs. So because nowadays the NGS is NGS cost is very low, and that leads to doing this GBS for large populations within the same species or large or different species, like large genome species which have a greater magnitude of diversity. So and this technique provides, as I said, a lot of SNPs for genotyping and for genetic analysis, for actually associating the phenotype with the genotype. And normally this technique is also widely used for germplasm characterization and various population studies. And the next step, which we go into the procedure of linkage mapping, is the linkage analysis. So as I said, linkage means is alleles from two loci segregate together in a family. So there is a formula that has been framed for recombination frequency. But before that, we would like to know what you mean by recombination fraction, actually. This is a probability of a marker and a susceptibility locus segregating independently during recombination. So if there is a 50%, this is the symbol theta that is being used for recombination fraction. So if the recombination fraction is like 0.5, there is no linkage that's happening. That means that the genes are located far apart, either on the same chromosome or on different chromosomes, assorting independently. But if the theta is less than 0.5, then it means that they are tightly linked together. And as I said, like, the recombination frequency means, like, you calculate the total number of recombinants divided by the total number of recombinants plus the non-recombinant products. I mean the crossover and the non-crossover products. So as I said, this is what happens inside the cell. So these are two homologous chromosomes, AB, capital AB and AB. When there is no crossover, then you get all the non-recombinant. You just get the parental segments as such after meiosis. Whereas when there is an exchange takes place during meiosis 1, this is called a single crossover. If there is another crossover happening adjacent to that, it will be called a double crossover. And so then you get 50% of these products after meiosis 2 will be recombinants, and 50% will be like parental gametes or the non-recombinants. So the frequency of recombination is being calculated from this, basically, and which says if the frequency of recombination is 1%, means it's just one genetic distance or one map unit. So which is genetic distance is measured in centimorgans, actually. And genes with these recombination frequencies less than 50%, as I said, they are located on the same chromosome, hence they are linked. So when two genes that undergo independent assortment, they should be on the different chromosomes, and they should be more than 50%, and they should be located on non-homologous chromosomes, or I would say, like, they must be far apart on the same chromosome, and this scenario is unlinked. So and all these kinds of like crossovers and the linkage happens like during meiosis, and it depends on the chromosomal arrangements during the metaphase 1 and the metaphase 2, and they could be in cis configuration and they could be also in trans configuration. So if you take these two different genes, because that's why there are two different genes having each of the copies, and they will be called a double heterozygote. So if they are actually, if these both the genes are on the same chromosome, they will be in cis configuration normally. And if they are in opposite, if they are on different homologs but on the same chromosome, they are in trans configuration actually. So when in my project, when I actually screened for recombination rate individuals, I found a lot of trans configuration in the trans configuration in individuals actually. So when I expected phenotype to be less than 20% or more than 20% or 20 centimorgan, I found a lot of these individuals like 80% and 90%, close to like 100%. So that means that these genes are located on different homologs on the same chromosome. Hence, they would occur in trans configuration. So it's a little bit points to actually keep in mind before we move on to the actual demo. So these are the things to remember. So recombination between the linked genes, they occur at the same frequency, whether in cis or trans configuration. But it occurs in the same frequency, but the thing is the overall value, I would say the magnitude or the physical value of that will differ actually, because as I said, cis will be less because the homologs will be on the same chromosomes, whereas in trans configuration, I mean the mutant alleles of both the genes will be on the same chromosome, whereas for trans configuration the value will be more actually, but the frequencies will be the same for the linked genes. So the recombination frequency is very specific for a particular pair of genes, and the more the recombination frequency, they are distantly apart actually. The genes are distantly apart, and no matter how these two genes may be, the maximum frequency of recombination between the two genes can only be 50%. So beyond the 50%, there is no actually, they are not linked anymore, and you cannot measure the recombination frequency. So, and certain things to remember, and the crossovers outside this gene region, that is the region, that is the recombined region or the region between the two genes, will be unaltered. Only the recombination happens between that specific segment, and any alteration happens only between the specific segment, between the two genes, and others are not altered. Others will be the same. As I told you, like as you can see in the picture, only that particular segment is altered and the others will be unaltered. And the result of double crossovers between these genes is indistinguishable from independent assortment of the genes. And because it's very hard to detect double crossovers between the two genes, you need an additional interval actually. You need 3 genes to actually detect double crossovers. And the double crossover, the percentage of double crossovers are very less in number. They are like less than 2% actually. So in gametes, they are just 1% in the 1% of the double crossovers happen. So crossovers involving 3 pairs of alleles specify the gene order or the linear sequence of genes. So this is something like a new, this is like, so it will be linearly arranged. If crossovers involve 3 pairs of alleles on a specific gene order, that is called, that will be on a linear sequence of genes actually. So this is something about some basic concepts I would want to tell. And if you have any questions, please let me know, because these are some terms to remember before we move on to the demos actually. So, I mean, it's quite a complicated concept, like between linkage and crossover. Often people confuse between the two phenomenon. And but I would summarize the fact that linkage is like, linkage happens between 2 genes that are tightly linked. They're very close to each other on the same chromosome, and crossover, and then crossover happens during meiosis, and independent assortment takes place on different chromosomes during recombination, and where genetic exchange of material takes place between the two like non-sister chromatids, or between the homologous pair of the non-sister chromatids, and genes that are located far enough, they are on the same chromosome, they undergo independent assortment, but linked genes, they fail to assort independently. So these are the things which we need to know. And whatever the genes are apart, or how far apart, the maximum frequency that it can go is only up to 50%. And beyond 50%, there is actually no linkage taking place. They must be very farther apart at that point of time. So this is something that this is in summary, in a nutshell, that I wanted to say. And what else? We can go for the demo tomorrow.
Divya madam, did you complete your presentation, madam?
Yes, sir.
Yeah. Bobby madam, are you ready?
Yes, sir.
Yeah.
Can you hear me, sir?
Yeah, yeah.
Thank you, Divya ma'am, and thank you, Sujan sir. It's a very nice session and very informative. And we will like to continue this tomorrow also. And I request Krupanidhi sir to take over.
Hello. Hello, participants and our guest speakers. We had a wonderful session for the past 2 hours on the first day, and it went on very well. Nearly 130 participants were there on Zoom, and our Sujan Mamidi sir is very nearer to our place, just 30 to 40 kilometers, where he has studied in Bapatla, and his native place is also very nearer to our VIGNAN'S University, that is Machilipatnam, around 60 kilometers from our place. And sir has given an elaborate account, and he has shown in the place where you were taking North Dakota University, where even in that snow, they were growing the flax seeds, canola, several of those useful materials which are commercially important plant products they are showing. Ultimately, they have come to the level of the genomics and several of those things, bio-entrepreneurship. Another important thing that that is missing in India is his institute is being supported by 40 industries, and they are paying rent, they are running everything, the maintenance of all those things. Such type of the institutions we need to have in India. Still they have to come up, and various things like variant discovery, epigenomics and plant genomics, and again the statistics and other aspects of which what he was mentioning is highly interesting to me. And in addition to that, he has also given 4 softwares like Karta Gene for the linkage map, QGene for QTL, and Tazzle for GWAS, and TextPad. All those things I request the participants to install in your computers from tomorrow onwards. Both Madam Divya and again our Sujan Mamidi sir will engage us in those type of how to operate, how to navigate this software tools. Once you get acquainted with this navigation of these tools, you will be thorough with the total understanding of the ecophysiological aspects and also the genomic aspects of the system that you are studying, whatever the case may be. Okay, that is with regard to the our Sujan Mamidi sir. Many thanks for having squeezed your time, and we are now in the odd time. We are now working in the daytime, in the morning, and Sujan Mamidi took out his time in the odd time and spent exclusively for us in this type of the workshop. Thank you, sir. And now our Divya madam is not new to our system of VIGNAN'S University. We have been closely interacting with her for the past 1 year, and she organized 2 or 3 with these online conferences. She's very good, and she's having a team of members from Coimbatore and UK. And madam, you have given a lot of your excellent, what is it called, track record. Particularly, you went through the Ratan Tata Foundation and later Commonwealth Fellowship, later Marie Curie research fellowship. And she also helped one of our students from BTech, who studied BTech by name Ravi, to get admitted in UK, Cambridge University. And she has shown total of the Cambridge University fundamental work related to the Arabidopsis and Meiosis, particularly leptotin, where this recombination signals are keep going on. And now I'll let you know by going through the total lectures of our Sujan Mohamedi and also Diva Madam. Those were the days we were studying the classical genetics, later genetics, later modern genetics, later molecular genetics, later developmental genetics, and now the recombinant genetics is coming into the picture, what we are seeing from the Divya Madam's work. Okay, so much progress is going on. So for all these things, one need to have the fundamentals, and she explained very vividly, lot of things about the linkage map and heterozygous, particularly while teaching this evolution, heterozygosity advantage. That's what in sickle cell anemia, if they both are recessive, means he suffers with the heterozygous. He suffers with the sickle cell anemia. If it is dominant, he suffers with the malarial infection. If it is heterozygous, means the fellow doesn't get the malarial infection, he will be a normal individual. Like that, heterozygosity advantage is spoken much in the evolution. So apart from that, Madam vividly explained the Mendelian law of dominance, law of segregation, law of independent assortment. Very vividly she gave the explanation for all those things. And again, the linkage maps, what Madam has done, and we have to understand still more. Of course, she has given excellently various aspects, particularly the theta values, how to calculate the theta, and molecular markers, AFLP, RFLP, SNPs, and all those things she did explain very well. Still, with the software tools that we are going to adopt from tomorrow onwards, we will learn more what Madam wants to make us to know. But one more interesting thing is she has done several samples with Arabidopsis. Arabidopsis itself is a small organism, plant organism. How many characters she has taken, how these characters have been linked to the genes is all quite interesting, quite interesting. All those characters need to be seen in this small organism called this Arabidopsis. So that's what earlier also I requested Madam to install Arabidopsis culture at our university. Still will continue our
Definitely,
interaction with
sir.
the
We will do.
yeah, we'll continue. So thank you, Madam, for all those inputs, and we'll continue this from tomorrow onwards. Thank you. Have a good day to you all. Thank you.
Thank you, sir. Thank you.
Shall we, Madam?
Sir, Sujan, sir, can you say a few words to wrap up?
That was an excellent presentation, Divya. You have provided all the basic concepts. From tomorrow, as we said, like we just want to stay, we don't want to feed you too many topics each day. We want to be simple as much as we can so that you don't feel bored and you will learn something out. And I have provided a link from my GitHub where you have you will have all the presentations and the data that we use for the workshops. And I just uploaded a presentation where it contains all the links, downloadable links for the softwares. They're all free of cost. So they're all open source. You can download from there. Make sure you have the prerequisites for each of those softwares installed correctly. And for tomorrow, we will be working with linkage mapping. So mostly we'll be working with Karta gene. I'm just pasting the links in the chat window where you can download the Karta gene. There are a couple of links and the instructions for installation. And I also provided a Google Forms link where you can submit your questions. Some of the questions me and Divya got answered in the chat window itself. But if we think if they are relevant, we'll try to get in a couple of slides tomorrow before we start the main workshop. And thanks again of all for joining. We'll take a leave for now and we'll join tomorrow with a demo session on linkage mapping.
Sir, can I end the meeting now, sir?
Yeah, sure.
Tomorrow we'll meet at the same time.
Thank you.
Yeah.
Thank you.
Thank you, sir. And
Diva ma'am, you continue.
No, just the recording is over, no?
Yes, yes.
Yeah. Just a few words after the meeting. I just thought I can talk few words with Sujan sir for tomorrow.
Okay, ma'am. If participants can leave, we can arrange the meeting.
Yes.
I'm requesting all the participants to leave the meeting.
Divya, we can leave too. We can have a different separate Zoom session.
Oh, sure. That's fine, sir.
Okay, sir. No problem, sir. Can I have the link actually?
I'll just send you an email.
Okay, sir. No problem.
I'm ending from my part, ma'am.
Thank you, Abraham. And tomorrow, I think we'll start by 9, right?
Yeah. Sharp. Every day start by 9.
Yeah, by 9. 8:45 will be there.
Yeah.
Thank you very much, and please post all the questions in the forms. We will attend to it later. I think I kind of gave some answers to the questions in the chat box, but for the rest, maybe please fill the Google forms. Maybe you can fill it every day, because every day you might have some questions. If anybody, you can put, or either you can put all the questions at once, or you can put clarify it then and there. I think clarifying it then and there would be sufficient, because we can answer them, we can prepare the previous day and come up with slides, or come up with an oral answer before you all. Thank you very much.
Okay, thank you.
Abraham, you can end up.